All right, I think we can start. Hello, everyone. So, yeah, we are continuing our conference with a great program and great content. And this time we have very special guest, who is Nitesh Turaga, and he's from Harvard Medical School, and he's core team member of Bioconductor project, and he's also Dana-Farber Cancer Institute of Harvard. He's a part of Dana-Farber Cancer Institute of Harvard Medical School. So today he's gonna talk about introduction to Bioconductor. Nitesh, stage is yours, and we are very happy to have you here. And I'm sure all the participants are as excited as we are, so we cannot wait. So please go ahead and start your talk. And we will be here on, and also questions will be coming through the chat during your talk. It's up to you. If you want, you can pick the questions and then just answer them during your talk, or you can leave them to the end. Okay. So yeah, at the end, Q&A also depending on you. If you will have still energy, we can also have the Q&A session. Yeah, but so we will be here in case of any technical issues. So we will be more than happy to help, and stage is yours.
Thank you so much. Thank you for inviting me to this talk. First of all, I'm really glad to be here. And I think this talk should be structured in like an interactive way. So if you guys have any questions in between, feel free to leave them up in the chat and we'll tackle them as I go. And we'll also have a Q&A session at the end. So it's no problem both ways. So today I'll be talking and giving you guys an introduction about Bioconductor and what it is, if you're not already familiar. And I'm guessing since everyone is at the YR conference, they're familiar with R at some capacity and the origins of R and so on, but I'll delve more into that. So I like to do this every time I give a talk. I like to put a really pretty picture and say where my slides are. So if anyone wants to follow along with the slides, feel free to like take a look at the slides while I'm giving the talk. So let me just give you guys an idea of like what Bioconductor is. So Bioconductor is an open source project. We build open source software in R, and our main goal is to produce software for analysis of data of current and emerging biological assays. We also focus on including a diverse, collaborative, and welcoming community of developers and data scientists across the spectrum. We have very strong open source background, and even going forward, we focus heavily on like open source and open development. And I'll give you guys more of an insight into like what I mean by that. Before I get into like the details, the nitty-gritty, let me just talk about like why anyone should use Bioconductor. So I don't know if you guys can see my mouse here, but I'm pointing at a couple of things on the screen. So in this triangle, at the lower edges of the triangle or the lower vertices of the triangle, you have basic genome biology, where you have all your biologists doing basic biology. And towards the other edge of the triangle, you have your data scientists who are working on like biological models with machine learning, simulation, high dimensional data science, and so on and so forth. And then in the middle of this lower edge, you have the biotech sector, which is coming up with like new sequencing methods, genome editing, so on and so forth. But the end goal of everyone on the lower edge of this graph is to move up to the top, which is some sort of like disease therapeutics and personalized genomic medicine. Bioconductor kind of helps everyone on the lower edge of this graph to join together, help them pre-process, analyze, annotate, and do many more things with their data and move up the sides of the triangle. And as we move up the sides of the triangle, we hit different areas of biology, like systems biology and population genetics and so on. So as you go up, the data size keeps increasing and the precision also needs to keep increasing. And Bioconductor kind of like fits right in the middle there and helps people who work in high throughput biology to like do different things to their data. I'll just pause here for a second and ask if anyone has any questions. Leave them up in the chat and I'll read them out. All right, everyone seems to be following so far. I'll keep going. So here I just want to talk about the timeline of Bioconductor. So Bioconductor and R started, so CRAN and Bioconductor, I should say, started in and around the same time. And they were also started by the same group of people, Robert Gentleman specifically. Bioconductor started in around like 2000, the year 2000. So if you follow along, there's 3 colors in this timeline: the green, which is the first decade, the blue, which is the last decade, 2010 to 2020, and then 2020 to 2030 is like we're in it, but it's also the upcoming decade. So we first started out with like microarray data analysis. Microarrays are a previous technology of like querying the genome. It was very limited. The data size was small. And then you can see that we developed a few packages for that, BioStrings and BioBase. And then the first commit happened around 2001 to our version control system. It was SVN back then. And then we had our first package, the website, the first Bioconductor conference. And as sequencing methods came up, we started writing packages for like DNA and RNA sequencing. And as we hit closer to like 2010, you can see that we started getting way more packages into our repository. There was a support website now which specifically answers questions about certain packages. R is really into the whole sticker ecosystem. So Bioconductor started making stickers. We have a scientific advisory board, a technical advisory board, and that started in this second decade. And we developed way more packages in the second decade. Our ecosystem size grew to almost 2,000 in this 2010 to 2020 decade. And new biological assays came up, single cell sequencing came up, and we developed different data structures to help people doing these sorts of analysis. And after this decade, around 2020, we started the community advisory board, and the data size of biology started increasing. All of these biological assays started developing bigger and bigger data sets. So in the last few years, we've been trying to move into age of the cloud. We started a great collaboration with Microsoft Genomics, and we started producing like different types of solutions on the cloud with them. And I think the next decade is going to be how we tackle this high dimensional data, how we store it, how we distribute it, how we compute on it. And I think the age of the laptop computer is like coming to an end slowly, especially with high throughput genome biology. So one more thing I have to add is like all these dates are approximate. It's a very compressed timeline. So take it with a grain of salt. Some of the dates might be off by like a year. We also have a very large industry footprint. Every time we host a conference, we're almost always sponsored by a host of amazing sponsors. They use Bioconductor in their data analysis day in and day out. You will recognize some of the names on here: Microsoft, Genentech, Novartis, Moderna, NanoString, and so on. So Bioconductor isn't just an academic venture. There's also a very strong industry footprint, which we have. As I promised earlier, I want to talk about this relationship between CRAN and Bioconductor. I'm hoping most of you are familiar with CRAN since this is an R-centric conference. So CRAN is also a repository. It was started slightly earlier than Bioconductor, but it's much larger and it's general purpose in nature. So you can write a package about pretty much anything and submit it to CRAN. Whereas Bioconductor is R packages, which are very specifically focused towards high throughput genomic data analysis. So if you want to write a package to analyze genomic data of any kind, you would submit it to Bioconductor. But it's not like these two repositories are completely independent. They are very interconnected and they're often used together. So you can use both CRAN and Bioconductor packages together on top of one another. You can have dependencies. If you're familiar with the dependency structure in R packages, you can have inter and reverse dependencies both between CRAN and Bioconductor packages. And this is often the case when we see packages submitted to Bioconductor. The one big difference between CRAN and Bioconductor is Bioconductor is heavily focused on interoperability between packages. And my talk will keep going back to this point of like interoperability and why it's specifically important to something like biology, and where we need like a little more structure in how we write our software. So just to focus on this point of interoperability, so these are these dependency graph clusters of the CRAN repository ecosystem and the Bioconductor ecosystem. And you can see that the CRAN graph, even though it has more packages, is more widely distributed, and they have many edge nodes in this cluster here. That's essentially saying that your packages don't really have to be depending on each other. They can just float in free space and do their own thing entirely. But Bioconductor necessarily doesn't allow that. Bioconductor is focused on one package being able to operate with another package seamlessly. So you can see that it's a tighter cluster. Even though the number of packages is like very large, it's almost like 2,000 today. It's a very tightly wound graph, and all packages are like interconnected with each other. Any questions so far? Okay, I'll keep moving. So we have a release cycle with both R and Bioconductor. And it's a reasonably complex release cycle if you're not familiar with it, but it's worth explaining and just giving people an idea of like how it works. So R itself is released on an annual basis. So if you see the top end of the graph here, you'll see that it goes from April 2020 to April 2022. These are approximate dates again. So you'll see that for every 1 year, R has one version coming out. So R 4.0 worked between 2020 to 2021. And from 2021 to April 2022, it's 4.1 and 4.2 and so on and so forth. We're still not at 4.2 yet. We're just getting there. But the idea is that R releases once a year. And if people were wondering if they should update to newer versions of R and Bioconductor, I think it's a really good idea to update because you get newer features. The accuracy of the programming language itself improves. There's many new benefits which you acquire by updating to the latest version. Bioconductor, though, releases twice a year. So for every one version of R, Bioconductor has two releases. So if you see the 4.0 April 2020 to May 2021 box right here, you'll see that for R 4.0, you have two versions of Bioconductor that work with R 4.0. So what exactly does that mean? It means that if you use, say, R 3.12 with R 4.0, that means that we guarantee as the Bioconductor core team that every package with 4.12 will work seamlessly with R 4.0. That's what that means in a nutshell. So if you're a developer for Bioconductor package developers are always required to use the version of R that will be associated with the next release. So you can see here that around the April 2022 mark, we have an upcoming release. So April 27th is the new release of Bioconductor, which will be 3.15. So developers for this upcoming release of 3.15 have been using R4.2 already. So that's what that means. And right now, at least till April 27th, 3.14 is the latest stable release, which works with 4.1. So this is a complex idea. I just want to make sure that everyone's following it. It'll really help in understanding both how R works and also how Bioconductor works, especially if you're a prospective developer or you're working in like genomics and you're wondering if you should update. The answer is yes. Okay, moving on. So the next topic, again, this is an introduction mostly on how to do things with Bioconductor. But if you want to install packages for Bioconductor, BioC Manager is a package which acts as this entry point into the Bioconductor package repository. Technically, this is the only package which is on CRAN. The idea of BioC Manager is it provides a safe way to install Bioconductor packages and check for any updates that may be available. But so once you install this package from CRAN, once you install BioC Manager from CRAN, this can be used to install packages from both CRAN and also Bioconductor. So if you're a new developer or user trying to get into the Bioconductor ecosystem, it's a really good idea to switch to BioC Manager to install your packages because it guarantees that safety and it also checks for updates. So if you've submitted a package to Bioconductor, right? So we have a build system, which is very unique in that it tests all the packages in Bioconductor together. This is coming back to my point of interoperability, where the reason for testing if all the packages work together for that version of R and that version of Bioconductor is so that we guarantee that all the 2000 packages which you install are in some way connected and built cleanly. So we build all of our packages every night. We build them on all three platforms, Windows, Mac, and Linux. This guarantees the robustness of our entire ecosystem with 2000 plus packages. This gives you like an idea of what our build system looks like. This is a public website again. So we focus on open development, open resources, open science, open everything. And you'll see that we have three platforms, Linux, Windows, and Mac. And you can see how many packages passed, failed, and so on. And these are the list of packages you'll see. And this is for BioC 315. So this is the current devel cycle. If you remember the previous, the release cycle graph I showed you. Any questions so far? Yes, the page's link is at the bottom. I think you might be able to see it, but I can copy it and paste it for you. if you would like. But my screen doesn't want me to for some reason. Oops. It's changing screens, but I'll keep that in mind and I'll give you the link to this page.
Mustafa already shared it, by the way.
Right.
So
Thank you. Yes.
we got it already. Great.
So after giving you guys a basic introduction of like what we do and how we do it, I want to talk about some resources where you can participate and engage with the community in Bioconductor. I think that one of the biggest strengths of Bioconductor is its amazing community. We have a wide network of users and developers on every continent, barring maybe Antarctica. So I highly recommend like getting engaged in the Bioconductor community. So first of all, we have the Bioconductor website. And these are just screenshots of the website, but feel free to explore them on your own. And the home just gives you like an idea of like what's coming up, like the biggest news items. So the Bioconductor 2022 conference in Seattle is coming up very soon. You can register for that. It's a hybrid session. I'll just talk more about it later on. And there's also help to, you know, like if you're a new developer or if you're a user and if you want some help, you can check out the documentation, the support site, what packages are available, what are the previous releases, what publications came out, and so on and so forth. And if you want to install packages, there's an install tab. And if you're a developer for Bioconductor, there's a very good guide on how to go about your package development in this developer section. We also have a support site. The support site is specifically for questions regarding one package. So say you're using a package like DESeq and you want to ask a question about like how to build your experiment with DESeq, or say another RNA sequencing data set, bulk RNA sequencing data set like EDGAR or something like that. If you post a question on the support site, it gets answered extremely quickly because there's a lot of people always looking and checking on the support site. Like I said, it's a very large community, and I highly encourage you to like make use of it. If you have any questions with any packages which are developed and submitted to Bioconductor. We also have a Slack channel with a lot of people, like almost like 2,000 or 3,000 people. We pay for it, but it's a very robust community. It's a great place to ask questions, interact with the community, just introduce yourself. And there's a link which you can click on here to join the community. It's free. Even if you're just an R user and you want to ask questions about R in general, I think it's a great place to ask questions about R. People are very helpful. They will answer all of your questions in an extraordinarily, you know, speedy time. Like they are always there. Like because we have this distributed community across every time zone, pretty much, people will get there, get your answers as soon as possible. I highly encourage you guys to join this. The next piece of resource we have is our GitHub organization. So we focus on open development. So even within the team, if we're developing a new package or if we're developing new infrastructure, we make sure that it's on GitHub and it's public. So we are able to take feedback from the community, from our advisory boards, from everyone, and we have this very social coding practices in place. So this is another resource, like if someone's looking to like use some of our products which are not on the Bioconductor repository, something which we use to like build our infrastructure and so on and so forth, it's a useful place to come to and like grab some code, grab some information readily. And all of our information is in an open source license, so you're free to use anything we have on our GitHub page. We also have the developer mailing list. So this mailing list is pretty much like a communication channel for package developers. Any announcements which we have from the core team, it's an extremely useful location to just communicate with a broader audience, I guess, and also with a generation which is very used to email if they're not already on Slack. So this is another resource to have in your book, just in case if you're not able to get the answer you're looking for on Slack. So now that you know what resources are available, how Bioconductor works, pretty much what it's about, you want to think about how to contribute. Like, okay, you're impressed by what I've said so far, and now you want to think about how to contribute a package. So the biggest way of contributing is submitting a package to Bioconductor. You submit a package on GitHub where you have, again, this idea of open development. We give you an open review and you have open build reports of what your package is doing and how it's working on all three platforms like Linux, Windows, and Mac. So we have a GitHub page for this. So I can write down the link here. I think I forgot to link it, but it's bioconductor.org. Sorry, github.com, bioconductor contributions. So I think that link will take you to the contributions page. But you can see that it gives you an idea of how to contribute a Bioconductor package and what to expect from the review. And each package submission is contributed as an issue in GitHub, which is on the left side of your screen. So you can see that right now we have 60 issues waiting for someone to review and get it into the repository. We have a team of around 10 or 11 reviewers right now. I'm included in that. And we take a lot of effort to review an author's package and help them improve their package and also give it a much wider audience. So if you are doing anything in the high throughput biology space and you're writing an R package for it, we welcome submissions on our repository. So this open review of packages specifically is a very popular idea in the open source world because it helps both reviewers and authors. And I want to give a shout out to GitHub here specifically because are there any specific, I'm sorry, I'm just going to pause here and look at questions here. Are there any specific requirements for contributing? So as long as the package has some big picture view of working on some data or annotation or anything from the high throughput biology space, you're free to contribute. There are specific requirements for contributing, and I can post a link here about the requirements. They're not very stringent though. It should be an R package or in the form of an R package or some data. And the next question is, are you looking for reviewers? Yes, I think they were looking for reviewers. I think there's a call that goes out for community reviewers. So everyone in the core team is part of the review group. And there are some people from the community who join the review group for a certain period of time as they get time and space in their schedule. But each package review takes about I want to say at least 2.5 hours of some detailed work. So the next time we look for reviewers, I will make sure to get the word out to people here. I don't know how to get in touch with you guys, but hopefully I can. Yeah. Hopefully that answers the questions. So I'll just get back to like why this open review system works so well. So I personally think that it takes away this idea of like hidden science and like hidden reviews, where the peer review concept is taken to the next step, where there is a code of conduct that is protecting both the reviewers and the authors. The package authors can engage publicly with the reviewers. The reviewers are there specifically to help build better packages, because we've been doing this like a long time. Like I've been writing R packages and working on Bioconductor for almost like 7-8 years now. So I have a little bit of experience like doing that. So if a new package author comes in, like I'm able to help them understand their package, understand the requirements of Bioconductor better, understand our build system, and just enable this full transparency. There's also a Bioconductor mentorship program, especially for people who are looking to submit new packages. So if you're a new developer and you have an R package in the form of scripts and you want to take it to a Bioconductor package level, we have the Bioconductor mentorship program. It runs on a yearly basis, where there's a few mentors. I think there's like 14 or 15 mentors. I'm not entirely sure. And each of us gets like 1 or 2 mentees, and we essentially help you get your package into shape for Bioconductor submission. And we impress upon you the coding strategies, which we've used for a long time, and help you understand them and develop your package using these robust coding strategies. So the current session is already underway. But if you sign up on the BioCDevel mailing list, or if you sign up on the Slack channel, you can watch for announcements for upcoming mentorship terms. This is an extremely useful resource. I signed up to be a mentor this year, and it's been very rewarding for me personally, and just to get ideas about new science, and also just looking at like how people write their code, helping a new developer along the journey it's fantastic. Again, highly encouraged to like take part in this program if there's any new developers in the audience. So now that we've spoken about resources, how to contribute, and so on and so forth, I want to talk to you guys about like the core concepts of Bioconductor and why we use the coding strategies we use. And this is essentially the meat of my talk, and you'll understand why this concept of interoperability is at the core of this Bioconductor philosophy. So I keep saying the word interoperability, like what exactly do I mean by that, right? So this figure kind of gives you an idea of like why we take it so seriously. So everything in the blue is a file format. And we try to like think about the sequencing ecosystem here. So if you go to the top left corner, you'll start with your sequencing files like the FASTQ file, you align it and you get a BAM file, and everything in the blue is a file format. And then you reduce these alignment files, the BAM files, into like a certain sort of signal. So if it's expression data, it's a sort of like count. It's literally just a matrix of counts, a single value for every gene and sample. And you can also get like differential binding, so binding sites and stuff, so peaks. You'll get VCF files for like variant files and so on. And everything in the pink here is a set of packages that will work on that specific file format. So you can go from your sequencing data, a FASTQ file, align it again with an R and using Bioconductor, reduce it to a specific file format, analyze these file formats with different sorts of analysis. So for example, if it's sequencing or expression data, you can do differential expression. If it's ChIP-seq, you can do differential binding, and you can see that there's different packages that work on it. And if it's genome-wide association studies, you have like other packages that work on it, work on VCF files and so on. And we take you all the way from right from the FASTQ file to integration and visualization of your data, to make sure you go from sequencing to actual results. And the major data processing steps are in blue, and then the relevant software packages are in pink. Any questions here? This is another important idea of interoperability again. Now I'll delve into like certain data structures, which we use to, encapsulate some of this data, so that I can talk to you guys about the robustness of the data structures we build. Okay, I'll wait for questions for another second. All right, moving on. So just to give you an idea of how robust our data structures are, I want to talk to you about this data structure called summarized experiment. So the summarized experiment data structure essentially fits into this, this counts matrix of sorts. So I'm just going to talk to you about like how we encapsulate that count data and fit all sorts of like relevant information around it. So if you look at the figure, right? So you'll see that there's an assay, which is in the middle. So think of every block of data here as like rows and column matrix. So you can see that in genomics, like all the samples are columns and the rows are certain features like genes. So the assay itself within this data structure is the actual count matrix. And it's a matrix-like object in R. So you can call dimensions, you can get dimensions of the matrix, you can call dim names and so on, matrix-like methods on it. And then the rows here are a certain feature or annotation on each row. So they can be genes, transcripts, you name it. It can be anything. It depends on like what the matrix is encoding, essentially. And then if you get row data or something like that, that can be p-values or log fold changes of each gene. And then the column data is usually the description of each sample, the sample name. And then you have metadata associated with it. The metadata is usually like unstructured metadata, and it gives you more description or like the models used to generate this data. And this is all one object. So the summarized experiment object, it sort of coordinates these assays, samples, regions of interest all together into one object. And we've written methods for it so people can interact with it. And people build on the summarized experiment object to build their own data structures. And this is how the interoperability works. So summarized experiment is a highly used data structure in Bioconductor. I'm going to talk about a couple of other data structures and tie them all together towards the end. So this is for assays. And now if you're thinking, okay, you do high throughput genomics, shouldn't you be accounting for like ranges or like sequences of some sort? So we have this data structure called RLE, or run length encoding. So the concept of like run length encoding here is encapsulated with an example of like a nucleic acid sequence. So before the encoding, so this is like a data compression method. I should say that it's a data compression method. And if the sequence is stored explicitly in memory, it's a very long sequence, but you can encode it, especially with consecutive runs of identical nucleotides. So for example, if you were to encode this sequence, you'll see that the A occurred once. So you get 2 bits of information here, the alphabet of the nucleic acid, and also how many times it occurred in that sequence. And then T occurs once, G occurs once, C occurs once, and then you have N 8 times. So that's how this compression works. And this is a very effective representation if your vector is very long. And this is often the case if you're encoding like string information, like genomic sequences, and also if there's a lot of consecutive elements of the same value. So repetitions are encoded very effectively. This data structure is called run length encoding. This is a very integral part of how we encode genomic ranges. So there's a package called iRanges that implements support for this class. And it's used in, so iRanges is a generic package, and genomic ranges builds on iRanges to support like the genomic range structure. And the code on this right-hand side is just showing you how to use the R code to use this run length encoding data structure. So the genomic ranges or G ranges object essentially represents a class or a collection of genomic ranges that have a single start and end site. So for example, if you have a binding site or a transcript or an exon, they have a single start and end site with some repetitions in between. So it's very useful to use this RLE or run length encoding data structure within it. So you can see that if you want to build a G-ranges class, and if you want to store like a feature, a genomic feature, you can use this RLE data structure to encode this data. Similarly, you can use iRanges, which encapsulates like a range, like a start and end site for a specific type of data set or like genomic feature. You can encode strand and so on and so forth. So again, the point I'm trying to make here is like the interoperability between different classes and packages is extremely important to Bioconductor. So this genomic ranges package builds upon like iRanges, and it provides support for like the BS genome functionality, our SAM tools, short read, our track layer, genomic features, genomic alignments, so many other packages. It's just incredible. And I want to tie together again to impress this idea of like interoperability, that you can combine G ranges and summarize the experiment and make another class, which is also very useful. So something like a ranged summarized experiment. So this is a child of the summarized experiment class. And all the methods which work on summarized experiment also work on range summarized experiment, because that's how inheritance works. And the fundamental difference between the two classes is the rows in a range summarized experiment are genomic ranges of interest. So for example, if you look at the airway package in Bioconductor, it's a combination of summarized experiment and G ranges. So if you load the data airway, it'll give you like a ranged summarized experiment, and the assays are counts, but the row names are ranges of a certain type. And just like any other biological data set, we almost always have like columns as samples and rows as a type of feature. And this airway data set is an RNA sequencing experiment, and it investigates human smooth muscle airway cell lines, which are treated with dexamethasone. It's a very interesting project, but I wanted to highlight the structure of the data itself. So this is an example of like how this interoperability works. And we have like coding strategies for different sorts of methods. So for example, if you want to import certain genomic data formats, like GTF files, bed files, and so on, you have our track layer, which you can use to import these files. If you want to import VCF files, you have read VCF. So there's already a bunch of stuff done for genomic scientists if they're building software, and you don't have to go back and write your own methods and classes. Similarly, for like SAM and BAM format, you have like our SAM tools, genomic alignments. For FASTA files, you have biostrings. FASTQ, you have short read. And this is like mass spec data. There's a bunch of like packages which help you import like mass spectrometry data. And we also have like not just importing, but like common classes. So for example, if you have a specific type of data set, like RNA sequencing data set, you can use the summarized experiment class to encapsulate your data. So this is like a rectangular feature versus sample data format. You can use it for microarrays, RNA sequencing, single cell, like has single cell experiment. Mass spec data has spectra, and then multi-omics data has like multi-assay experiment. And if you have like genomic coordinates which don't exactly match all too well, you can use like RAG genomic coordinates. And GRanges does genomic coordinates. Yeah, so there's so many different coding strategies and like common classes built for people who are using this already. But say you're one of the people who needs to build off of something that's already available, and your data needs a new representation, you can carefully design an S4 class. I'll talk about the S4 class structure again a little bit. The S4 class system is very integral to Bioconductor. We build most of our like popular data structures in the S4 class system because it allows typing, and it's just a very robust class structure. It's safe, it's very robust. But if you do build a new data representation, it's in your best interest to build a class which is carefully designed. You can help other package developers with your hard work. You've already done the hard work of building this class, so you might as well promote reusability of that hard work. And if you write a really good vignette, users will use your package, and they'll also be able to use other packages which build off of this class. So I just want to talk about the S4 class system here, just to give you guys an idea of like how Bioconductor builds some of its like core data structures. So the S4 class system is implemented in the base package in R called methods. It's heavily used in Bioconductor. And on the left side here, you'll see you have a class, S4 class 1. It contains two slot names, slot name 1, slot name 2. And these two slot names are restricted to have slot type 1 and slot type 2 type of data sets, respectively. And this class also has a validity rule. So there's a set of rigorous like validity rules that check for the integrity of the data each time an object is updated. So this is how an S4 class system is built. So that's like the base idea of an S4 class. And then I'll talk about inheritance here. So if you want to build a class that works similar to the S4 class, but has an extra slot, then you can inherit from S4 class 1. So this S4 class 2 object contains S4 class 1, has everything S4 class 1 already has. So slot name 1, 2 are inherited. Similarly, the validity rules from S4 class 1 are inherited, but you can add a new slot type. You can add more validity rules. And this right here at the very bottom is showing you how to create new objects of a certain class. Can you guys give me a yes or no if you're able to see my mouse on the screen? Because I've been...
Yeah,
Okay.
now we can see actually. Yeah, it's visible.
Okay. Okay, thank you. Yeah, I've been pointing, and I just want to make sure that you guys are able to see it. And then on the right side here, you have, I want to like just impress upon you like the idea of like method dispatch polymorphism in the S4 class system. So these are like common concepts in like any object-oriented programming concepts, like very general to many other programming languages as well. But the way it works in like the S4 class system is a little different. So you write a generic function which doesn't have like a function definition in it just yet. It just has a signature that it takes an object of type X. And then you have to build two methods. So if you want your generic, this S4 generic one function to work on object one, you have to write a method for it. And then you have to write another method if you want it to work on object two. So the definitions of both these methods are going to be different. And when you call the process, when you call the S4 generic one function on, either object one or object two, it'll make that decision of like which method to use based on the object which is given as input. So this is this idea of like method dispatch and polymorphism, this idea that you have a generic function which works in two different ways depending on the object which is given as input. Any questions so far? This is very generic object-oriented programming concept, but this is how all of the Bioconductor class systems are written. And if you wanted to write a new class system, this is how you would do it essentially, right? Using S4 classes. So just to give you like a little bit of a background. So the RLE data structure connects to the I ranges. So let me just read the question here. These newly developed Bioconductor packages are new solutions to the problems we face when dealing with genomic analysis. I wonder how, so let me just read the question out loud properly. These newly developed Bioconductor packages are new solutions to problems we face when dealing with genomic analysis, right? I wonder how they decide what to develop even when there are 2000 plus packages. So it usually depends on which type of biological assay they are dealing with. So if it's a new type of assay, let's say something like spatial transcriptomics, which is just coming up, you would have to write a new class structure and just contribute to the repository in such a way that other people who are going to work in spatial transcriptomics can use your base package and then build different sorts of analysis packages on it. Does that answer the question? I'll take that as a yes. Again, feel free to post any more questions you may have. So like I was saying before, so the RLE data structure is in this package called S4 vectors, and iRanges builds on this RLE concept, and then genomic ranges builds on iRanges. Summarized experiment can use GRanges. Range summarized experiment can use summarized experiment and everything before it. So there's this core idea of like interoperability and also just being able to use other people's work before you, which is very critical to like how science works in general. And we need to make it easier to do that. And that's what we're striving for. Trying to put together like this very robust class structures, taking input from the community and so on. All right. Yeah, so these are just like advantages of like using something like this. So most of the useful data structures are already written for you. I'd like to think that we are pretty much on the forefront of genome biology and high throughput biology in general. We have a lot of community members who work in industry, in academia, and they contribute to us quite a bit. So we are on the forefront of genome biology. And if you're looking for a data structure to either write a new package or, or if you want help, like just make sure to check before rewriting something. Most of the times it's almost always ready for you already. And these are very robust. They're peer reviewed, they're discussed, they are interoperable with other data structures. So it's useful to check before rewriting. And each package in Bioconductor, this is my favorite part about Bioconductor, has a very detailed vignette on how to use it. So for more strategies for developers and documentation, please click this link. It'll take you to, the developer site where you can look at the guidelines on how to contribute a new package. I'll leave this in the chat. How to contribute a new package, what guidelines are required, like how to structure it, some basic coding strategies for you guys, how to use version control and so on. It's very useful. And there's also like a coding practices section here. So it'll talk to you about like what common methods and classes are needed, robust and efficient coding styles. So you need to vectorize your code, make it as efficient as possible, pre-allocate and fill. So there's some really robust like guidelines for you to follow here, and how to write efficient code for Bioconductor. I'll leave this in the link as well. Okay, so I just want to touch on like the next decade of the timeline I showed you guys earlier. So from 2020 onwards, Bioconductor is focusing heavily on containers and cloud computing. And I want to give a shout out to Dr. Erdal Kozgun. He's been an invaluable partner in like helping us like go forward with this and get to where we're going together, I guess. So we build Docker containers in Bioconductor for people who are worried about installing packages, installing system dependencies, and so on. Like if you're familiar with Docker, it's a very useful product, which will help you get going with Bioconductor directly. So you can use this Bioconductor Docker image. There's a devel image and there's a release image. And it has all the system dependencies you would need already. It'll work on all three platforms, Windows, Mac, and Linux. Yep, that is correct. So there's a question there saying, where can we find these Docker images? Can we find them on Docker Hub? Yes, you can. So if you click this link, it'll take you to the Docker Hub location where you can pull these images and use them. And since this year, we've also been like providing this feature of binary package installations. So not just for CRAN, for Bioconductor as well. So you can install on average a Bioconductor package or a CRAN package 7x 7 times faster than you could on your local machine. So if you are using a Docker image, like you get many advantages on top of what your local machine has to offer. So you can install pretty much any Bioconductor package seamlessly. You get an RStudio front end. If you're an RStudio user, it's the community edition, but still works very well. And we have a new release coming up very soon, April 27th. So the tag release 315 will be available as soon as the release comes out. So like I was saying, these images are available on Docker Hub. So you can click this link, go to Docker Hub, and you'll see that the latest image right now is the release 314 because the new release hasn't happened yet. But as soon as that happens, you'll see the new tag and you should be able to use it. Any questions here? Okay, I'll go on to the next. We're almost done with the talk for what it's worth, if you guys have spaced out. But we have a code of conduct in Bioconductor. Since it's an open source organization, we have a code of conduct, and the current version is as recent as July 27th, 2021, it was updated. The idea of this code of conduct is to promote collaboration, have a kind and welcoming environment, and promote diversity and inclusivity of people from like all sorts of backgrounds. So I welcome you guys to join our community because it's a safe space. Any question is welcome, and you're welcome to contribute, join, participate, however you want. There's another question here. While running Bioconductor on top of Docker, should I copy my R scripts to my Docker container, or is there an easier way? There is an easier way. So if you go to the website and you click on, so there's this Docker link here. So if you click on that, they'll show you like how to use your Docker containers efficiently. It'll also show you like how to deploy your Docker containers on cloud systems if you want. But you can mount a volume. It'll show you the exact commands to do so and run the Docker image. But the easiest way by far is using this Docker Compose link. So if you download, or if you pull the repository from GitHub, you can just write this Docker Compose up command and it'll do everything for you essentially. So yeah, so maybe that should get you going. So all your local machine, everything on your local machine will be mounted on to YAML files are ready to use, or should we configure them? Yeah, the YAML files are pretty much ready to use. So I can show you the repository for Docker. Where is it? Right, so we have this GitHub page. So you can copy this or clone this repository. And if you say Docker Compose up, it should pretty much work seamlessly. So you can see that, so this is the devel image right now. So you'll see that your volume will be hosted, and you can add like another line here which says mount a local volume with RStudio Home or something like that. But yeah, you might have to add like another line here, but it'll work nonetheless. Yeah. But if you just want to mount a certain volume, you can use another line just like this, run it interactively as well. I think that should have answered that question. Yes, our studio UI is being hosted on localhost 8787. That is correct. And the username is our studio, and the password is something you can set as however you want with an environment variable. Any more questions? I guess we can expose another port. Yes, you can expose it to another port as well. Yes. You just have to change the, I'll just go back here. Oops. Nope, that wasn't it. So I forget where I was, but yeah, so if I were to like go back here to Bioconductor, bioconductor.org, say Docker, and then running the containers. So instead of like 8787, so this port exposing command here, you can just say a different port, whichever is open on your machine, and you can expose it. Yes. And even within the Docker compose file, you could do that. Yeah, so you can change it to whatever you want, whatever port is open on your local machine. Does that answer your question? Yeah. Thank you. Yeah, it is very straightforward. Yes. Right. So I just wanted to like finish up my talk by saying that we have the BioC 2022 conference coming up from July 27th onwards to 29th in Seattle. It's hybrid, so it's in person and virtual. So if any of you are interested in like attending in person or virtually, registration is open. So please join us. It's a very diverse group, which some of these numbers show you from like the last conference. And if you want to join, like the website is given below, so feel free to join. We also have conferences in Asia, Europe, at least once a year, not during the pandemic, but they're starting up again. During the pandemic, they've been virtual, but yeah, please join and enjoy what we have to offer and participate. I want to say thanks to my amazing team members and also Dr. Vince Carey, Martin Morgan, and Raffaele Rizzari. They make a lot of this possible. And if you have any questions, feel free to ask. This is my email. You can email me questions. We can do a Q&A session right now. You can also email your questions to Bioconductor. And there's a Slack channel. Let me see if I can copy the link and post it in the chat so that people can join the Slack channel. I think that is it.
Amazing, Nitesh. You know, that was great. That was great. And also thanks for doing this session interactive, taking the questions whenever they appeared. And it was, I think, very, very valuable. And personally, I really enjoyed especially the Docker part. And last year, we also had a pleasure to have Erdal during our conference. And Erdal was mentioning about also Docker part. But this time I also had a chance to learn other part of it as well. And it was really cool.
Thank you. Yeah.
And, you know, especially when with Docker, it's like very straightforward. You don't need to worry about any type of dependency. So everything is maintained by Bioconductor. Just pull the new and up-to-date image and just go ahead.
Absolutely, yes. So I want to say that the Docker images are also hosted on Microsoft Container Registry for like, if someone is using Azure, like they can use that. So MCR Bioconductor. So if you just Google MCR Bioconductor, like it'll show you, you know, the registry hosted by Microsoft. And if you're using Azure, like this is, even the Docker part of it will become quicker.
Yeah.
But pulling the image will become quicker. It's more robust, so on and so forth. Yeah.
Is it possible to have it also for ECR for AWS?
Yes. So you can use the same image on AWS as well. Elastic Container Registry. Yeah.
Yeah.
Use it
Okay.
pretty much anywhere. So we don't host the images there, but you
Mm-hmm.
can use it there
All
as
right,
well.
that's great.
And we have like many more Docker solutions. Like we have like a Kubernetes based cluster you can launch. There's many amazing cloud solutions coming up. So when I mentioned binary packages being available, so from the new release, so release 3.15 onwards, so when you say BioC Manager install within the Docker image, you should automatically get like binary packages for Bioconductor as well. So everything will be that much faster on a Docker machine as opposed to a local computer, which isn't configured.
Absolutely, yeah. And also everything is on the top of Linux, so
Yeah,
without any pain. Yeah,
exactly.
yeah,
Yes.
exactly. That's great. So just one question maybe from my end. So imagine that I'm using the Bioconductor on the top of Docker. So I also, created some scripts and I run some analysis and I stored the results and everything is stored inside of the container. Should I have some persistent storage in case of having the new update of this Docker image? So do you have any easy way to do this? Do I have to deal with that or not really?
No, not really. So I'll show you why. So github.com. So for myself, so this is a little hack I use for the Docker images myself. So I have a personal page, build Docker. So if you look at the Docker compose file here, it actually just connects my...
Mounted to your local file. Okay, local directory. Yeah.
It just mounts to my local file system.
Yeah.
And I have like a Dropbox which backs everything up in this by default.
Okay. Yeah, makes sense. Yeah.
So that's like the easiest solution for me. So I can link this if any of you guys want to add it. You obviously have to modify that path here, but like if it's available, it'll work. Yeah.
Thanks.
And when you update the Docker image, you don't have to. The one thing that will happen though, so if you update the Docker image, it'll create a new directory where the latest version of the packages are installed in that new directory. So you don't have to like actually worry about it. So for example, like all the packages which are mounted will go into 3.15, but if it was like release 3.14, they would go into release 3.14 directory. So there's a really good separation of like the versions of packages and so on and so forth. Yeah.
All right, that's great. That's great.
Any more questions? I'm happy to take any question. Feel free to ask. Yeah.
Yeah, maybe just one more question from my end as well. As Bioconductor, do you provide some, because when you want to do some calculations, so it's always good to run them on the cloud, on the top of GPU, and now at the moment we are talking about TPUs and so on and so forth. So there are some free and open resources for that, but they're not really stable. As Bioconductor, do you provide something like this to the researchers? So I don't know, some access to any type of cloud and where we can run Bioconductor, for example.
So if you want to run Bioconductor, so we don't provide a cloud directly.
Okay.
You have to pay for it. at some level. But there are like solutions which are more reasonable than others, I guess. So using these Docker images, you can launch. So you can launch pretty much anything on like Azure container instances, for example. And we have instructions for that on how to launch a Docker image on an Azure container instance. And you can start and stop your container instance and like work with it very seamlessly. But we're hoping that we can get like some access to like a public cloud in the US to potentially provide the kind of cloud solution you're looking for. But the solution we have right now is called the Anvil. So it's a Google Cloud-based environment. It comes with the same Docker images which you're using right now.
Cool.
But it's also going to come on Azure pretty soon. But you can use the same Docker image as a cloud environment on this Anvil environment and like use it on Anvil platform. And you get access to like data sets, protected data sets, so on and so forth. Yeah.
Okay, that's really cool. Thank you.
Yeah.
I think there are no more questions. So just maybe encourage our participants to ask questions once again. So you can ask your questions, you can text them, or just feel free to raise your hands and we can unmute you and just ask your questions directly to our speaker. Maybe let's, Nitesh, give like a few more minutes to our participants. Maybe they will have some questions in their mind.
For sure, yeah. So what is the distribution of like, I guess like R to Bioconductor users in this group of people? That'd be an interesting question. Like, does everyone use Bioconductor? Are people familiar with it, or are they just learning?
Yeah, maybe our participants, you can just say yes or no to the chat. We can run like a very simple survey on the fly.
Yeah.
Okay, so obviously Erdal is using.
Okay.
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 Basic level, no, one no. Okay. Okay.
Perfect. Yeah.
Fantastic.
Yeah.
All right, so I think there will be no more questions. Nitesh, once again, really, really, really thank you so much. And thank you for taking your time. So you spent your time with us, you shared amazing information with us. And once again, thank you so much. And so hopefully we will see each other again, but definitely
Yes.
we will be we will be in touch with you to learn even more about Bioconductor from your end. And once again, thank you so much.
Yes, please feel free to contact me. Thank you once again for having me here. I really appreciate it. It was great talking to everyone. Thank you.
Perfect. And then thanks a lot for joining today. So today there will be one more session together with Christian Zielinski. We will be talking about deep learning. So we will have a break, and so we will start at 5 p.m. Central European time, and see you there. And thanks a lot.
Thank you. Bye-bye.
Bye-bye.